Okay, so ladies and gentlemen, what I'm going to do now is I'm going to give you some information. And if some of you like to write down notes, you can think of this as notes, okay? And you can also, if you want to get this information again, I'm going to put this recording up for you in the classroom. So you can also just go back and look at it and listen to me again. And what this is going to be a diffusion. Come on. Diffusion pre-lab notes. That's what this is, okay? So you're going to have, you're going to be doing an ed puzzle, not this class, but next class, we're going to have you look at an ed puzzle, and this one is going to count as a lab, because this is an actual demonstration that I did, a live demonstration where you're going to see me doing a laboratory experiment in front of you, and then questions are going to pop up, and you're going to need to answer them. And in order to answer them, you're going to need a little information from me first to explain things, which I'm going to do now, and then you're going to need to just watch it as a YouTube. Just watch it. This is going to be your homework. You might even be able to start it before we leave, okay? And write down what you see me doing. Write down anything that you think is important. Think about it a little bit, and then next time we come together, when you actually look at it for real, and it's a real ed puzzle, and it's going to count as a lab grade, that's a big grade, you'll have a really good idea of how to answer the questions as they pop up in that ed puzzle. So the first thing that I want to do is define a couple things for you. So first of all, let's define diffusion. Diffusion is the simplest type of cell transport. Can anyone chat to me what they think, how they would define diffusion? I know you learned this in the 7th grade, maybe before. What is diffusion? They are so sleepy today, Mr. Bouchard.
Oh, they are.
You are. I'm usually getting some answers here. No one wants to bite on this? Okay, when water moves from different concentrations, that's getting around it. Not just water, though, right? Lots of things can diffuse, and it's true that they move from different concentrations. Yeah, okay. Ramey's got a lot of it in there. I'm going to pull that together. Yeah. Oh, yes. You're talking about all the words I'm going to define today, so good for you. Diffusion is a process where molecules move from from high to low.
Secure, I sent you a message really quick while Ms. Murdoch's typing. Secure, I sent you a private message just to give you a heads up to hear. If you look up in the message.
That's what diffusion is. That's what diffusion is, okay? And then I like what Ramey said, and I actually had written down the same things here. I think we should talk about what a solution is and what its parts are. So a solution, let's talk about that for a minute, is a mixture made of a liquid solvent, which is almost always water, and solutes. Things dissolve in the water. Okay, there we go. So a solution is a mixture of two things. A liquid solvent, right? Solvent. And solutes that can be dissolved in that solvent, right? And then there's a word that that's up here and that Ramey also used called concentration. So let's talk about that for a second. Is diffusion only from high to low? Diffusion by definition is always from high to low, Ramey. That's very, very good point. If it's going the other direction, it's no longer called diffusion. It's called movement against a gradient, which is a form of active transport. So that's coming up next class. That's coming up in a few classes from now. It's really good, Ramey. You're way ahead of me. But let's talk about that word concentration, because that is not an easy word. What does concentration? What does concentration mean? Right? Concentration means it's a measure of how how much solutes there are in the solution, right? So for example, if you have, 5 grams of sugar per liter of water, right? Versus 10 grams sugar per liter, right? Which one of these, the 5 grams of sugar per liter or the 10 grams of sugar per liter, is more concentrated? Which one? The 5 or the 10? Yeah, the 10. So the 10 grams sugar per liter, there's always grams per liter or amount per volume, right? Amount of mass per volume usually. That's going to be more concentrated if you have less grams of sugar per the same liter, right? Okay, so this one is has a higher concentration of sugar, which is the solvent, right, than this one. Okay, so just playing with the words here a little bit. All right, another word that I want to tell you about is the word okay, there's going to be things coming up. I think I'm just going to let this go. All right, so I'm going to draw a picture for you now to bring this all together. What if I have a beaker? Okay, and I've got a solution in there that is divided by this semi-permeable membrane. And you guys know what semi-permeable is because you just saw a pear deck on it, right? So this semi-permeable membrane right here permeable membrane, right? Now we know cells have semi-permeable membranes with the phospholipids and the proteins, but semi-permeable membranes can also be man-made. There are scientifically created sort of these polymers, these plastic membranes that are made that are not made of phospholipid bilayers and special proteins. That's something that only life can make. But human-made semi-permeable membranes are sort of plasticky things that just have little tiny holes in them. And it's just a matter of how big the holes are. Are these holes here big enough for, they're almost always big enough for water to go through, but they might not be big enough for other bigger things like sugar molecules to go through. Okay, so now given that story, this semi-permeable membrane right here, it allows, well, I'm going to have it allow both water and glucose can go through it, okay? But larger things like sucrose, which is a disaccharide, it's bigger than glucose, can't go through it. All right. So let's say that we have on this side, we have a whole bunch of, I'm not trying to draw a face, a whole bunch of glucose molecules over here. It's a very high concentration of glucose molecules. And over here, there's just a little bit. That's it. That's all, right? So over here, maybe we've got the 10 grams per liter over here, and over here we have only 1 gram per liter. Okay, so the concentration of glucose is higher over here than it is over here. And according to what I've told you, this semi-permeable membrane will allow glucose to go through it. So tell me, if I let this beaker sit over time, what's going to happen? What's going to move and which way? Come on. on, guys. What's going to move? This is all glucose molecules over here, right? The solute over here is glucose. Those little marks, that's all glucose. And it's very concentrated on this side. Okay, so what I'm asking is, over time, if I let this beaker sit, right, what's going to move and which way is it going to go? Water and glucose can go through this membrane. What do you think is going to happen over time to these concentrations here? Here. Yes, very good. Okay, so that's all I wanted. Yeah, okay. So the glucose here is going to go through to this side, isn't it? Until what's going to happen is, yes, Ethan, you're brilliant. My gosh, so smart. It's going to keep moving until it reaches equilibrium. Exactly right, Ethan. You are right with me. Yes. So the glucose is going to move to the left. The glucose will move to the left. Good for you. Glucose will diffuse to the left side until equilibrium is reached, right? Until both sides have the same concentration, okay? So this is now in a state of equilibrium where you now have, and wow, that was cool. I didn't expect you guys to come up with that, but yeah, until the concentration of glucose is the same on both sides. So it will be 5.5 grams per liter on this side in the end, and it will also be 5.5 grams per liter of glucose over here, right? Exactly right. Okay. So here's the thing. I told you that the glucose could go through the membrane. So in the movie that you're going to watch, I'm not going to tell you what can move through the membrane and what cannot. So I'm going to erase this. Don't worry, I'm recording it, so you can go back and get it anytime. So now that you know all those words and we pushed all those words around, what if you have, and you will, you have a situation, which is the exact situation that you'll see in the movie, the YouTube epic movie, I'm the star, where I'm going to put a solution inside a bag and close up the bag tied off so that it kind of looks like that. And inside the bag, there's going to be water. There's going to be water as the solvent, and then there's going to be a glucose in there, and then there's also going to be some starch in there. So you're going to have those three things are in the solution inside the bag. And then outside the bag, out here in the water around the bag, water is also the solvent. And there's going to be some iodine. Okay. So that's the start of the experiment. And the question is, we don't know how small or large the holes are in the bag. We know that there are tiny little holes in the bag. You can be pretty sure that water can get through them because water can get through just about anything. But can the glucose move, right? Can the glucose move? Can the starch move? And, can the iodine move? That's the question. Those are the questions I'm going to be asking you. Is what is the permeability? Right? I'm asking you, what is the bag permeable to, right? What can go through it? And not. Right? So molecules have sizes, right? Some molecules are bigger than others, some are smaller than others, and you might remember a little bit about how big glucose is versus starch, and iodine is just an ion with a charge to it. Right. Okay. So here's some more clues. I'm just going to give you a few more clues. I'm not going to give you answers here because I want you to figure them out. But do you remember the chemical indicator lab and the fact that one of the things in this picture is a chemical indicator? Which one of the substances that I've typed here in this picture is a chemical indicator that changes color when it hits something?
Ooh,
Which one of those
that's a good
substances
question.
in there?
Do you want them to put it in the chat, Miss Murdoch?
Yeah, go ahead and put it in the chat.
All right. Let's see it, guys.
Is glucose a chemical indicator? Is starch a chemical indicator? Is iodine a chemical indicator? Remember, a chemical indicator is a chemical that changes color when it encounters a particular molecule. Are you guys really just that tired?
Oh, think about it, guys. Just take a chance. Nobody's going to get mad if you don't get it right. I get half my life wrong.
Yes! Yeah. Benedict's solution is a chemical indicator. That's beautiful. I didn't even have it up here, but I'll put it up. So Benedict's solution is one. That is one that in my video. And the other one that's in this picture is iodine. Both of those are chemical indicators, right? They change color when they are in contact with something specific. Benedict's solution changes color in the presence of glucose. Yeah. And iodine, yes, changes color with starch. Exactly. That's right. Now you're right.
Oh, good. Good job, Ramy.
Right. Okay. So now I just want to make sure you understand. Now you know what, right? So Benedict's solution is a chemical indicator, and it changes color with glucose. Iodine is a chemical indicator, and it changes color with starch. Okay. That's it. Those are the only two you need to know. Yeah, that's okay. Benedict's and iodine are all you need for this lab. And understanding that Benedict's is the test for glucose, iodine is the test for starch. So right now, do you see in the picture that the iodine and the starch are separate, right? So right now, the iodine is not going to be able to turn black with the starch because it's out here, this solution out here. It's out here in this solution out here, the iodine. And the starch is inside the bag, away from it and separate from it, you see. Okay? So right now, at the beginning of the experiment, before anything starts moving, there's no way for any color change to happen, right? But if you let this sit overnight, and I did, some very interesting things happen. And that's what the video is going to show you and going to ask you questions about that. Okay? So that is the end of the pre-lab, and I want you to make sure that you have all that information. Now your homework, let me just turn this off. Your homework then is going to be to go and watch YouTube. I'm going to show you where that is.